Once the polymerase has escaped promoter and initiated transcription, it shifts into the elongation phase. So first was the formation of pre-initiation complex, then the transcription was initiated, and once this passed the initiation phase and the promoter was passed, then next step was the elongation phase. This transition involves the polymerase II enzyme shedding most of its initiation factors, such as the general transcription factors and the mediator. So all the factors which were recruited during initiation, they are mostly shedded when it enters the elongation phase. In their place, another set of factors is recruited. Some of these, such as TFIIS and SPT5, are elongation factors, that is, the factors which stimulate elongation of transcription. Others are required for RNA processing, because as the RNA is synthesized, it is being processed side by side. The enzymes involved in RNA processing are like several of the initiation factors recruited to the carboxy-terminal tail, CTD tail, of the large subunit of polymerase II. So here you can see different factors. This is capping enzyme, this one is capping enzyme, and this is the splicing machinery, and this one is polyadenylation and cleavage factors. So these are different factors which are now recruited during the elongation phase. In this case, however, the factors favor the phosphorylated form of the CTD. So before these factors are to be recruited, the CTD should be phosphorylated. Thus, phosphorylation of the CTD leads to an exchange of initiation factors for those factors required for elongation and RNA processing. So once the CTD tail is phosphorylated, it results in the exchange of these or replacement of these factors. As is evident from the crystal structure of yeast polymerase II, the polymerase CTD lies directly adjacent to the channel through which the newly synthesized RNA exit the enzyme. So this CTD tail lies adjacent to this RNA exit channel. The CTD tail is also very long. It could potentially extend 800 angstroms from the body of the enzyme. That is about 7 times the length of the rest of the enzyme. So it is very long tail. Together, these features allow the tail to bind several components of the elongation and processing machinery and deliver them to the emerging RNA. So these factors are delivered to the RNA molecule, which is a newly synthesized RNA. Various proteins are thought to stimulate elongation by polymerase II. One of these, the kinase P-TEFb, is recruited to polymerase by transcriptional activators. Once bound to polymerase II, this protein phosphorylates the serine residue at position 2 of the CTD repeats. So at every position 2 of the serine residue in the CTD tail, it causes the phosphorylation. That phosphorylation event correlates with elongation. In addition, P-TEFb phosphorylates and thereby activates another protein called SPT5, which is also an elongation factor. Finally, TAT-SF1 Yet another elongation factor is recruited by P-TEFb. So TFIIS1 is then recruited, which is also an elongation factor. Thus, P-TEFb stimulates elongation in three separate ways. SPT5 is comparable to the bacterial elongation factor NASG. Indeed, this is the only universally conserved transcription factor across all the three kingdoms of life, that is, bacteria, archaea, and eukaryotes.